Hello everyone, myself Vedika Gardi. So today we are going to talk about a sequencing method called as ion semiconductor sequencing, also known as ion torrent sequencing method. So first of all, what are sequencing methods and why are we using them? Sequencing methods refers to technique which will determine the exact sequence of a nucleotide or a basis in a DNA molecule. So, these sequences have biological information encoded in them. Upon knowing the sequences, we will understand various functions of the gene or other parts of the genome. This study will form a base of biological research which can be applied in various fields like biotechnology, forensic biology, medical diagnosis, even finding of mutations and any genetic alteration in the molecule. There are various type of sequencing method like Sanger sequences, whole genome sequences, and ion semiconductor sequencing is one of the method in them. So generally, whenever a nucleotide is incorporated into the DNA strand, a hydrogen molecule is released as a byproduct. This same principle is implied over here, wherein this method detects the release of hydrogen ion when a base is incorporated into the template strand which is to be sequenced. This detection is based on a sensor. So there are various micro wells in this device beneath which a semiconductor chip is present. This semiconductor chip has a ion sensitive field effect transistor sensor. Whenever a base is incorporated and a hydrogen ion is released, this sensor gets triggered by that release of hydrogen ion. This refers that the reaction has occurred. The reaction will change the pH which is detected by the sensor, which in turn is given out as a voltage which can be measured digitally. There is no fluorescence, optic or anything involved in this method. A chemical reaction will directly give out a electronic signal which is detected by the computer. So the mechanism is as follows. It is sequencing by synthesis method, which means the sequences is done by synthesizing the DNA. This method will follow the normal DNA replication method, wherein a complementary strand is built based on the sequence of the template strand. There are microvels which will contain template DNA strands. Then DNTPs will be fed in this microwell. If this DNTP is complementary to this template strand, this will continue further growing into the complementary strand. For example, if there is a cytosine present on the template strand and complementary strand will have a guanine, this two will be matching with each other. This will form a complete nucleotide, GC base, and this will release a hydrogen ion. This hydrogen ion will trigger the ion sensor and will give out signal, and a computational data would be formed. If there are two nucleotides incorporated, then voltage will be double, which will be given out. There are various steps involved in this process. First is the fragmentation of the DNA. So, as there are multiple microvels present, we first need to fragment the DNA into various fragments. We can use chemical or any enzymatic methods to fragment the DNA, due to which each microvel will have a different fragment of the DNA or have a different template of the DNA. Then addition of adapter sequences. Adapter sequences are nothing but the complementary bases which are complementary to the template to be sequenced. These are 20 to 40 base pairs long. This will form a complete nucleotide when added into the microwell. As you can see in the picture just right next, there is a guanine present onto the template strand. When cytosine from the DNTP or the complementary strand is coming and joining to here, an H or the hydrogen molecule would be released. In the picture next, the hydrogen ion is detected by the sensor. This sensor will give out signal as a voltage and a computational data would be formed. There are various applications for this method. First is small genome sequencing, wherein various small genome like a mitochondrial genome. Rapid sequences for mitochondrial genome can be done, which can be used for disease research and biodiversity assessment. Amplicon sequencing can be used for discovery of somatic mutations in a complex sample, wherein the DNA molecule is mixed with germline samples. So, from this complex samples, also we can analyze our choice of the DNA. In transcriptome sequencing, fast identification and characterization of various interesting transcripts can be done. Fourth is targeted DNA sequencing. So, by using this method, we can completely focus on to one specific set of the gene or analyze them for a specific mutation or any changes. Along with its applications or advantages, there are various limitations also for this method, like the it has short read length when compared to other sequencing methods. Also, this system is unable to enumerate long repeats. So, example, when a homopolymer repeat like an adenine is repeated continuously, then the system is unable to enumerate this much long repeat into the data. The throughput is also lower than that of other throughput sequences. These are some of the references which I have used for referring my topic. Thank you.